Leventhal here, and I've got another video lecture for you. And today we are going to learn a little bit about genetics. We'll call today's lecture Genetics 101. And what I'm going to do in our discussion today is I want to talk a little bit about some of the vocabulary in genetics. I'm also going to introduce you to was often referred to as the father of genetics, and that is Gregor Mendel. And finally, I would like to do a demonstration on how to do a basic Punnett Square. And how this connects with meiosis. So hold on to your hats, and here we go. Have you ever noticed that when you look at people, that some people have variations that are different from the characteristics that you have? So clearly we note that there are a wide range of variations in hair color and eye color and height and hair texture and skin color and so on. And in my graphic here, I'm sharing with you three variations that we often see in people. For instance, some people have what are referred to as free or detached earlobes, while others have attached earlobes. In contrast, believe it or not, some of you who cannot roll your tongue may not know that others can roll their tongue. And the opposite is true, that I'm fairly certain that many of you who are rollers did not know that there are non-rollers. And then finally, it's interesting that some people have what we would refer to as a hitchhiker's thumb, and others have what we refer to as a straight thumb. And these variations exist in so much of our biology. Biochemically, we can talk about people who are lactose intolerant versus those who can drink milk as adults. And so these are variations, and these variations reside in our genes. So we talk about genes, which are located on chromosomes, which is made up of DNA. And if you have had me in class, you know that I like to draw a graphic that looks something like this. And that is, your DNA has many genes, and a single gene, or your sequence of DNA, codes for proteins, and these proteins give you your phenotypes, which are your physical characteristics. So if I back up a page, we can say that some people are phenotypic for attached earlobes, while others are phenotypic for free earlobes. And you can be phenotypic for straight thumb versus hitchhiker thumb. And the difference between these are essentially based on proteins that are either coded for or not made, depending on your trait. And ultimately, whether you have hitchhiker's thumb or not hitchhiker's thumb, is based on a gene which comes from a DNA sequence. So DNA, small pieces of DNA code for genes. Both of these are essentially instructions for making proteins. And these proteins give you the physical traits that we use to define our characteristics, and we call those physical traits phenotypes. Well, the person who began the biological study of this process is this man, Gregor Mendel. And Gregor Mendel was a monk in an Austrian monastery. And he was actually a very educated man, educated in physics. He actually worked under a guy named Doppler. You may have heard of Doppler with the Doppler radar. He also studied plants. And when he ended up in the monastery, he inherited the monastery's garden. And that garden had a huge record of plants that were grown throughout history. And one of the plants that they grew a lot of in this monastery were pea plants. And so Mendel got into studying peas because he noticed that peas had a lot of variation. He found that peas could have purple or white flowers. He found that the peas themselves could be green or yellow. He also found that the pods could be green or yellow. And there were a number of other traits, like the height of the pea plants, as well as whether the flowers grew axial or out of the sides in a bushy fashion versus terminal along the top. And he defined these traits, these patterns. He said, hey, there are factors, that was his word, and we now use the word genes, that control flower color or pea color or pod color and so on. So genes here are determining color. Genes are determining height. Genes are determining position. And then he said within these genes there are alternate forms and he called those alternate forms alleles. So we could talk about the purple allele versus the white allele that is coded for by the gene for flower color. We could talk about green peas or yellow peas, the two alleles for pea color, and so on. So alleles are alternate forms of a gene. Therefore, here we could say that tongue rolling is a gene where you could be a roller, one allele, or a non-roller, the other allele. So let's look at Mendel's first and simplest experiment. Mendel actually describes in his research the trait for flower color. I'm going to use pea color here. And what Mendel did was he grew green pea producing plants and yellow pea producing plants. And what he found is when green producing plants reproduce with themselves, that is, green plants, green pea plants bred with green pea plants, they always produce babies with green peas. Similarly, when he took yellow pea-producing plants and crossed them with yellow pea-producing plants, all of the babies produced yellow peas. And he said that these plants were true breeding. And the word that you might be more familiar with, rather than saying true breeding, is the word purebred. He said they're purebreds. Green pea-producing plants crossed with green pea-producing plants always produce green pea babies. And yellow pea-producing plants crossed with yellow pea-producing plants always produce yellow pea babies. And he asked the question, hey, what happens if I were to take one of my green true breeding plants, so one of these guys, and I were to breed it with a yellow true breeding plant? So all of a sudden he's taking purebreds for different traits and he is going to cross them. So I'll give you a moment. What do you think he's going to get? Drum rolls, anyone? Well, it turned out that when he did this, all of the babies had yellow peas. And Mendel was kind of puzzled. What happened to green? That's a really good question. It was a question that drove him. So he was like, all right, how can I find out what happened to green? So Mendel then decided to take these yellow hybrids and he decided to allow them to inbreed. Actually, they self-fertilize. He basically took the pollen from a flower and mixed it with its own egg. So he kind of forced it to have sex with itself. And this was a good way in order for him to see what might have happened to green. And when he did this, he found that he ended up with a mixture of yellow and green. And in fact, the mixture worked out to be a ratio of about 3 yellow pea-producing plants to 1 green pea-producing plants. So let's look and think about what type of conclusions were made as a result of this. So if we look at our first genetic cross, Mendel came to the conclusion that yellow is a dominant trait. He said it was dominant because this is the visible trait in the hybrid. In contrast, he defined green as a recessive trait. Now, recessive does not mean bad, and dominant does not mean good. Recessive simply means the trait masked in the hybrid. So it's the trait that kind of disappears temporarily. So now let's try to figure out how these numbers came about. How did we end up with a 3-to-1 ratio when we did what's called a hybrid cross? So where did this ratio come from? Well, Mendel knew nothing about meiosis, but all of this is based on the process of meiosis, and I'm going to do a quick review of meiosis with you before we look at how to put this stuff into the Punnett Square. So let's review meiosis for a moment. And I know that this is a topic that we've discussed already. Meiosis is a specialized type of cell division. It's specialized because it doesn't occur in all of the cells of your body or most of the cells in your body. It only occurs in that group of tissue that we refer to as gonads, because this is where the gametes are produced. And so we produce our sex cells in the process of meiosis, and these sex cells are haploid because they contain half of the chromosome number. So let's look at what might occur in meiosis, and let's look at the trait that's coded for by the letter A. And so I would like to point out to you that we are looking at a cell where the diploid number is equal to 4, and the blue and red chromosome are holding genes for a trait that we're representing by the letter A, where the red chromosome has a big A, and the blue chromosome has a little a, and we could say that big A, little a is hybrid for a trait. And so what's going to happen? Well, we're going to go through S, and in S, each of the chromosomes is going to replicate, and we're going to have paired chromosomes. And now we have the X-shaped red and the X-shaped blue and the X-shaped gray and the X-shaped orange, but still this cell is diploid. It's got 4 chromosomes. And we're going to go through meiosis 1, and what's going to happen in meiosis 1 is a big chromosome is going to go into one cell, and another big chromosome is going to go into the other cell, and a small will go into one cell, and the other small will go into another cell, and all of a sudden we have now made our haploid cells. So now n equals 2, and then we're going to go through meiosis 2, and in meiosis 2, I want you to notice that the chromatids separate. And these final 4 cells, well, these are going to be gametes, and so if this is a male, each one of these would be a sperm. And so I want you to note that 50% of the gametes have big A's, and 50% of the gametes have little a's. And so now we can look at what's going on in a Punnett Square, and I'm going to take you back to our first Punnett Square. And I want to remind you what we learned here. We have a purebred green pea-producing plant breeding with a purebred yellow pea-producing plant, and we produce a crop of just yellow peas, and we've decided that those yellow peas are dominant because that's the color we see in the hybrid. And so, in the world of genetics, we do something special, and that is we are going to assign a capital letter for the dominant trait, so P for peas, and a small letter for the recessive trait. And we actually use a two-letter code, and the reason why we use a two-letter code is that you have two of the same chromosomes. So here, two big chromosomes, each big chromosome holding the letter A. So we can say that this yellow pea plant is big P, and the green pea plant is little p, and the reason why we use the same case is that this defines a purebred for that trait. The same letter says, hey, we are purebred for yellow, big P, yay! And here we are purebred for green. So little p, woohoo! And so when we set up a Punnett Square, what we do is we're going to take one parent and put it along the top, big P and big P. And then the other parent we're going to put along the side, little p and little p. And what this Punnett Square is doing is it is simulating meiosis. It is separating the homologous pair. And so we could think of this little p as being inside of a sperm, and this little p as being inside of a sperm. In contrast, the big P is in an egg, and the other big P is in an egg. And when we do a Punnett Square, we're saying, hey, what's going to happen when this sperm and this egg get together? Well, we get big P, little p. Well, what about when this egg and this sperm get together? Big P, little p, and big P, little p, and big P, little p, and all are big P, little p hybrids, all are yellow. So now when we go to the next cross and we have our big P, little p hybrids, and we say, okay, what are we going to do? We are going to cross big P, little p with big P, little p. So what happens? Well, we'll put a big P in one and a little p in another. So again, this could be a sperm. This is a sperm. 50% of the sperm have big P's. 50% of the sperm have little p's. That is just like what this picture of meiosis illustrates. And we are doing a hybrid cross. So again, big P, little p here. These would be eggs. And so what do we get? Well, we have one big P, and we learned here that big P is purebred yellow. And we get, whoops, one little p, and we learned that that is green. And then we get big P, big P, little p. And we know those are hybrids. And we already know that the hybrids are yellow. So let's take a look and do an accounting. We have yellow, yellow, yellow. That's 3. And we have only 1 green. That's a 3-to-1 ratio. The other thing that we could do is we could describe the ratio of allele pairs. Ratio of allele pairs. This is referred to as our genotypic ratio. And here we have 1 big P, to 2 big P, little p, to 1 little p, little p. Well, that's it. That's our intro to genetics and how to set up a Punnett Square. More on this when I see you in class. Have a great day.